Thanks everyone for attending on a different day from our normal CIC lectures. I think our speaker needs no introduction, but I'm gonna do that anyways because that would be rude not to. So our speaker today is Dani Bassett, who's done a lot of work on kind of examining complex systems, so they could be social systems, brain systems, and so on, and how these things interact. And there's just a tremendous body of work that Dani has done that a lot of us are kind of reaching into for a lot of the types of work that we do. Dani is the J. Peter Skirkanich Professor at the University of Pennsylvania, with appointments in the Departments of Bioengineering, Electrical and Systems Engineering, Physics and Astronomy, Neurology, and Psychiatry. They're also external professor of the Santa Fe Institute. And Dr. Bassett is most well known for blending neural systems engineering to identify fundamental mechanisms of cognition and disease in human brain networks. So thank you very much for coming to the talk today. Very much looking forward to it. And I'll give you the podium.
Great. Thank you. Thank you so much for having me for today. What I wanted to do over the next couple of minutes is talk about quantifying mental effort in a brain network economy. And I know that that's a mouthful. So what I'm going to do is to separate out each of these pieces so that we can kind of understand what I mean by mental effort, what I mean by a brain network economy, and why the two should go together. But before I do that, I want to start by talking about biological systems a little bit more generally. So one of the beautiful things I think about biological systems is that their functions are very complex, and those complex functions are made possible by networks along which various items of interest can travel. So for example, there are networks at the gene scale, the scale of molecules, the scale of cells, the scale of organs, the scale of the entire human, the scale of multiple humans in social networks. And in each of these cases, there are pathways between units along which information can travel, or nutrients can travel, or signals can travel. The brain is just one example that I'm particularly obsessed with. And the way that the brain works as a network is that there are individual pieces of the brain that are distinct in terms of cytoarchitecture, in terms of electrophysiology, and in terms of functions. And those individual units support the variety of cognitive processes that humans enjoy. But they do that by passing signals to one another through this network of interactions. And so what you're seeing here on the left-hand side is an image of the brain and the patterns of structural connectivity at the large-scale human brain. And then what you're seeing in these little windows is sort of a blow-up of tracks that are estimated from diffusion imaging. And those are the tracks along which information can travel. Now, importantly, what I am interested in is trying to understand how this network that exists in every human brain supports the kinds of activation profiles that we observe. So as we think about this system, we know that different regions of the brain can be activated, and then the brain moves from one activation state to the next, to the next, to the next. Well, how does it move between these activation states? It moves between them by allowing information to flow along these tracks. And so the pattern of network organization here, the pattern of structural connections here, constrains where activity can flow, and therefore constrains where the brain can go. And that is really the motivating observation behind what I'm going to talk about today. So over the next couple of minutes, I'll separate my comments into three main sections. In the first section, I'll be talking about the role of structure in determining the cost of activity flow. I'll first start with a case study of how TMS can flow through structural connections to exert changes on activity dynamics. And then I'll try to generalize from a single tract and a single study to a whole brain pattern of structural connections. In the second section, I'll try to build a more formal mathematical account of this network economy using control theory. And then in the third section, I will use that idea of a network economy to provide an explanatory account of mental effort. So let's dive into the first section, the role of structure in determining the cost of activity flow. So the first question I want to ask in this section is, how does structure constrain the flow of activity? And I'm going to give an example in the context of transcranial magnetic stimulation. So this is a paper that was recently published in Science Advances, I think in 2022. The first author is Valerie Sidner, who's a graduate student in neuroscience at Penn. And then the senior author is Desmond Othis, whose work is commonly in fMRI and TMS. And then Ted Satterthwaite, who is in the Department of Psychiatry. So the way that they developed this study is by recruiting 45 healthy participants and performing an interleaved TMS fMRI session, in addition to acquiring diffusion imaging data. So the diffusion imaging data was one where we processed it using QSI Prep, which is a pre-processing piece of software. And then from that, we built a whole brain tractogram. And then from that, we quantified the fiber density between two regions of interest, the one that was being targeted by stimulation and the one that we wanted to affect using stimulation. So then the TMS part of it was one where we applied TMS to the ventrolateral prefrontal cortex, which I will refer to as VLPFC in the rest of these few slides. And we know that this region has a known role in emotion regulation. The actual structure of the experiment was an interleaved single pulse TMS with the functional MRI. So the first question is, how does stimulation to VLPFC activate the target of interest, which for us was the amygdala? And so what you're seeing here on the left-hand side are individual stimulation sites for all 45 participants in the study. And then here what you're seeing is the magnitude of the TMS-induced changes in the bold signal when the TMS was applied to the VLPFC, and what we're looking at is changes in amygdala activity. So first note where the 0 line is here. So you can see with TMS, some individuals have a decrement in the activity of the amygdala, and some individuals have an increase in the activity of the amygdala. Our next question was, how big was that change in evoked activity? And that's what we're showing over here on the right-hand side, where above the 0 line means that the evoked response magnitude was larger in the amygdala than in these other regions. That includes the caudate, the putamen, the hippocampus, and the thalamus, and the pallidum. And then anything that is below this 0 line is where the activity evoked by transcranial magnetic stimulation was smaller in the amygdala than in these other regions. And you can see that's only true for the accumbens. So what's important here is to note that TMS elicited a sizable decrease in the fMRI bold response in the ipsilateral amygdala, and that those evoked response magnitudes were greater in the amygdala than most other subcortical structures. So that's indicating some degree of spatial specificity. Now, why am I using this study as an example of structural constraints? Well, our next question was, how are those individual responses explained by the structural connection between where we focused the TMS and where we were trying to target, which was the amygdala? So we studied this pathway between VLPFC and amygdala, and we quantified the fiber density between those two regions. So on this middle panel, what you're seeing is the evoked response, or the change in bold magnitude after TMS. And then along the x-axis is an age-corrected fiber density between where the TMS was targeted and where we wanted to affect. So you can see a nice positive relationship between these two variables, indicating that when participants had a high fiber density between those two regions, the TMS had a bigger impact on the amygdala. And when fiber density was small, the TMS had less of an impact. So what that indicates is that the structural pathway between these two regions and individual differences in that structural pathway are explanatory of individual differences in response to the TMS. Now, one last point I want to make about this study is that importantly, there are some individuals I pointed out at the beginning who have an evoked response that's larger, so greater bold magnitude after TMS or with TMS, and others had a decrement. And we see that those are also explained by the fiber density. So that's this panel here on the right-hand side, where along the y-axis is the left amygdala signed TMS evoked response, and then along the x-axis is the age-corrected fiber density. What I'm showing here is that higher fiber density was associated with a bold decrease in these negative evoked response individuals. That's this dark line here. And then bold response had increased in those with a positive evoked response, and that's this light purple here. So what that means is that the density of this fiber pathway was explanatory both of an increase in activity and a decrease in activity, depending on how that person responded in general. So what this suggests is that, or what this offers is a structural explanation for individual responses to TMS. In particular, it provides evidence of amygdala engagement by TMS, highlighting stimulation of this VLPFC amygdala circuit as a candidate for potential treatments for transdiagnostic psychopathology. But more broadly, it indicates that targeting these cortical, subcortical structural connections might enhance the impact of TMS on subcortical neural activity, and by extension, subcortex observed behaviors. So that's a first piece of data in support of this notion that structural constraints can impact the flow of brain activity. But importantly, that's only one link in a system that has many, many, many links. So the second question that I want to ask in this section is, how does a whole structural network, not a single structural pathway, but a whole structural network, how does that constrain changes in brain activity? And for this, I want to start with some intuition before I build in the mathematical framework. So in working through this intuition, I'm going to use these images that indicate, for example, brain states. So here is one brain state. It is a vector of activity across neural units, where a darker color indicates more activity and a lighter color indicates less activity. So I'll call this vector a state. It's just a pattern of activity across the brain. Here's a second state and here's a third state. Those brain states can do work for us. They can represent objects, they can represent events, they can represent experience, they can represent emotional states or cognitive states or concepts. And what I'm interested in asking is, how does the brain move from one of these states to another? Importantly, I'm interested in how this pattern of structural connections between regions constrains where high activity can move. And so that determines how easy it will be to get from this state to this state, or from this one to this one. To show you how that works in practice, I'm going to give you a sort of toy example. The top one is the one I'm going to focus on first. This is where we have an easy transition between two states, and then I'll focus on the difficult transition. So let's focus our attention on the top of this slide first. There are two pieces of this picture. One is the state that I've just been describing. So this is a state where the first 5 units in the toy example are active, and then the second 5 units are not active. That state sits on top of this highly modular network, where the first 5 units are densely interconnected with one another, and the second 5 units are densely interconnected with one another. And then there's only one line in between. This state is really easy for this network to get to, because I just need to send a generic excitatory signal into the first module and a generic inhibitory signal into the second module. And because there's only one line of connection between, it's easy for the activity to stay on the left and for the inhibition to stay on the right. It also means that it's easy to transition from this state to this one. So notice that where we're going to is where anything that was on initially is now off, and anything that was off initially is now on. To enact this state transition, we can capitalize on the structure of the network and the segregation of connectivity, and just inject an excitatory signal into the right-hand module and an inhibitory signal into the left-hand module. And then again, use the fact that there's only one weak connection in between them that will keep these two states of dynamics relatively separate for a relatively long period of time. Not forever, but for some period of time. Now I want to contrast this scenario, which is an easy scenario. I only had to inject 2 control signals into the system to move it into the state I wanted it to go to, with this difficult scenario at the bottom. So this difficult scenario is one where the network is the same. It's a modular network, but the state that we are in is really different. So in this case, the state is the first unit is off, the second unit is on, the next unit is off, the fourth is on, etc. This state is actually very difficult for this network to get to because you have to be injecting excitatory or inhibitory signals into every single node in the network to get it to where you want it to be. There's no way of you have to actively fight against what the network is supporting. And then if you want to take this system and move it to a second state, again, you have to fight against the network to do that. So here what we're doing again is anything that was off initially is now on, and anything that was on initially is now off. To enact this state transition, I need to inject an excitatory or inhibitory signal into every single node in the network. So I have to actively fight. I have to squelch this one and activate this one, etc. So I'm saying that that state transition is difficult because there's a larger number of control signals that need to be injected into the system. There's a greater spatial extent of control signals that need to be injected into the system. And there also, I'm not showing this here, but there can be time varying constraints. I have to inject those signals in a particular time in order to allow this state transition to happen. So when I'm saying that a state transition is easy, what I mean is that there are few control signals, they're relatively simple, they're spatially constrained, etc. When I'm saying a transition is difficult, it means there are more control signals required over a greater spatial extent and with more temporal precision. So the idea then is that when the brain is moving from one state to another, there's a particular cost. There's an energy, how much control needs to be injected into the system to allow this state transition to happen, given the network structure that that participant has in their brain or that patient has in their brain. So what we'd like to do is to have a way of thinking about these changes in energy, how much energy is required, and how much control input is required and where, and that provides us with a cost for state transitions. And that cost is what defines the economy of the system. A system can be more economical if you can move between states with less cost. And a system can be less economical when you're moving between states that have a higher cost. Okay, so that brings me to the second part of my remarks, which will be focusing on building a formal account of this network economy using control theory. And in doing that, I'm going to try to walk through 3 points. The first is addressing the question, what is a network? What is network control theory? The second is using network control theory to probe controllability, particularly in the context of psychosis. And the third is using network control theory to understand the cost of brain state transitions. After I finish the second point, I'll move on to the third, which is the network economy accounts of mental effort. So let's start with the second section, building a formal account of network economy using control theory. So what is network control theory? This is a theory that's been developed in the context of electrical and systems engineering with input from computer science, from physics, from mathematics, and from statistics. And the basic idea is that when you have a complex network dynamical system, it's interesting to ask how that system responds to exogenous inputs and what kind of exogenous inputs we need to inject into the system to move it into a desired state. So I'll walk through this image, which again is a toy example for building intuition. Here you have a network with 4 nodes and 4 edges. The inputs are U1 and U2 that are injected into nodes X1 and X2. The inputs themselves, notice, are time varying. So this suggests that it's possible to inject input into one of these nodes in a time varying way, with more input now, a little less next, and then more, etc. Now the question is, how does this network respond to those 2 inputs? On the right-hand side, you're seeing a quantification in a state space, where perhaps the initial state of the system is where all nodes, X1, X2, X3, X4, had an activation of 0. So the system was sitting at the origin. When these signals are being injected into X1 and X2, that pushes the system into a different place. But it doesn't just impact X1 and X2 because activity flows from each node to others in the network because they're all connected. So the activation of all 4 units is altered. And that's indicated by the flow of this line to some final desired state in this 4-dimensional space. This approach has typically been applied to the study of things like the power grid or mechanical systems, air traffic control systems, and robotics. But over the last couple of years, it's become clear that it has a lot to teach us and tell us about how nervous systems work, and in particular, how we can understand control mechanisms in neural systems. So how do we use network control theory to try to understand the brain? the first thing that we need to do is to have an understanding of the network. So that's what I'm showing you here. Here is the pattern of structural connectivity in a human brain as estimated from diffusion imaging. We apply a parcellation to this data so that we then have a weighted adjacency matrix where each element in this adjacency matrix A tells us something about the strength of connectivity between nodes I and node J. That could be the fiber density, like I just showed you in the TMS study. It could be the number of streamlines, could be the mean diffusivity, it could be any measure of the microstructural integrity or volume or curvature of something about that structural connection. But in addition to understanding what the network is, we also have to have a model of dynamics of the system. What I'm showing you here is the very simplest model of dynamics that you could write down for a brain. And then there are ways of expanding this, but for intuition's sake, I want to walk you through the very simplest version. So here what I'm showing is that the state of the brain X, so that's the pattern of activity across all neural units, at time t plus 1 is equal to the state of the brain at time t, so that's the moment ago, times the adjacency matrix. So if you close your eyes and ignore the BU part, and you just focus on the first two pieces, what that is saying is that the current state of the brain, that activity can flow through structural connections to explain where the brain will be next. That's all it's saying. It's just saying the brain has a state, the activity can flow through structural connections and explain where the brain will go next. Then when you add in this final term in the equation, that is providing exogenous input from somewhere else in the brain or from somewhere outside of the brain. So U is the control input that's injected into kappa regions at a particular time t. And this matrix B is a matrix that typically has ones along the diagonal that indicate where the inputs are being injected. So this part of it just says what is coming into the system to perturb it away from this simple diffusive dynamics. Now, this equation that I'm writing down, as I said, is the simplest you probably could write down. It's a linear model without noise, without time variance of the adjacency matrix, without any nonlinearities. It's a very, very simple one. So a question that has come to the fore over the last couple of years is this particular very simple equation one that still provides us with some intuitions in neural systems? And I wanted to point to a recent paper that was just published in December. It was led by Irfan Nozari, who was a postdoc in my lab between me and George Pappas, who's a control theorist at University of Pennsylvania. And Irfan led this study to examine the utility of linear models in explaining brain dynamics. So this is a paper that just came out and provides some really important validity to this model applied to neural systems, particularly when modeling fMRI signal, but also intracranial electrocorticography. But once you have a model of the dynamics, whether it's the simple one or something more complicated, the next question is, what do you do with the model? How do you use that to try to understand the network economy in the brain and how that may impact cognitive function? So one of the things that you can do is to try to understand control strategies that are possible for the brain system. Two control strategies that have been studied a lot over the last couple of years in neural systems are modal controllability, and that's the capacity to push the brain into a part of the state space that's far away from where it's currently sitting. And then average controllability, which is the ability to push the brain into more local areas of the state space. What we found in really early work, 9 years ago now, is that regions of the brain that can push you into a distant space on the energy landscape are often found in frontal parietal cortex. So in regions of the brain that are important for executive function, inhibiting inappropriate behavior, complex decision-making, working memory, cognitive flexibility, and other related functions. By contrast, the regions that are better at controlling or pushing the brain to more local states nearby where it currently sits are those in the default mode system. So what that suggests is that different pieces of the brain are structurally predisposed to enact different control strategies with very low effort. And in particular, the default mode regions are those that can enact the control strategy of pushing you to most of the places you need to go nearby, whereas regions in the frontal parietal cortex are structurally predisposed to be able to push you into a state that's far away. That's particularly interesting when we're thinking about the role of these regions in task switching and in cognitive flexibility. So that capacity for frontal parietal regions to push you into different states is something that may be structurally explainable. What I'm showing on the right-hand side is that these two kinds of controllability also change over age when executive function is becoming more and more common for the individual to engage in. So here, this is work from the this is data from the Philadelphia Neural Developmental Cohort in collaboration with Raquel and Ruben Gur and Ted Satterthwaite and others in the team. On the right-hand side, you're seeing the age of the individual from 8 years to 22 years of age. And then every data point here is a different person. Along the y-axis is the modal controllability, and along the x-axis is the average controllability. We see a positive relationship here such that as individuals are developing over this period of time, more controllability of both kinds is present. So that suggests that the brain can engage in these different control strategies more easily with time and with development. One of the questions that we had, though, is after this study was, well, what is actually changing in the white matter or in the structural connectivity of the brain that explains this increase over development? And this was work that was led by Lyndon Parks, who was a postdoc in my lab and has now started his own lab at Rutgers University. And he suggested or kind of reasoned that there may be multiple drivers for changes in controllability. There can be importantly local drivers for these changes and also non-local drivers. What do I mean by a local driver? Well, a specific region of the brain, depending on how many connections it has, can exert different sorts of control on the rest of the system. So this would be the sort of number of connections that are coming out of a region, so its degree or its strength is one way in which controllability can be changing. But there can also be non-local drivers, so something much broader beyond the single regions and about how regions are interconnecting in larger circuits that is explaining changes in controllability. So Lyndon hypothesized that these two potential explanatory factors were present in the brain, but in different areas. And in particular, he hypothesized that regions of the brain that are unimodal, so in motor cortex and in visual cortex, their local structure is going to be changing in a way that predicts the changes in controllability. By contrast, regions in transmodal areas are ones where the more global or wider scale circuit organization is going to matter. So to test this hypothesis, what he did was to plot the correlation between the controllability, which is here AAC, and the strength. So that's the local connectivity coming out of a region. So here's the correlation between those two variables as we move from unimodal regions to transmodal regions. And what you can see is that in unimodal regions, the control capacity is well predicted by local organization, just the strength of that region, how many connections and how strong they are coming out of the area. And by contrast, once you get to transmodal regions, the correlation between controllability and strength is very weak. So there's something about the more global circuit organization that is explanatory of the control properties of those regions. So this is a way of thinking about the complexity with which transmodal regions are enacting influence on the rest of the brain. They're doing it not necessarily because the local organization is different, but because the wider or global organization is different. And this became particularly important in explaining individual differences in psychosis symptoms. So this is work that was published in Biological Psychiatry recently. And what Lyndon did was to build a predictive model where some variable X was used to predict some variable Y. Our interest was using controllability to predict positive psychosis spectrum symptoms. We controlled for age, for sex, for brain volume, and for in-scanner motion. We used 100 repeats of 10-fold cross-validation, where each repeat used a different random split of the data. And what you can see here is the RMSE, where lower values are going to be better. Again, with the unimodal to transmodal axis present in the x-axis. So here what you can see is that the model provides these really robust estimates of prediction performance differently for the average controllability versus psychosis than for the strength versus psychosis. What we were able to find is that the average controllability of transmodal regions is predictive of psychosis, but the controllability of unimodal regions was not. And there was not as good or a significant prediction from strength. So what that means is that particularly in transmodal regions, their controllability, which takes into account wider circuit organization, is predictive of psychosis spectrum symptoms, particularly positive symptoms. So that suggests that the complexity of the local organization around these areas changes or impacts what kind of influence those regions can have on the rest of the system with behavioral ramifications. So that's one thing that we can do with network control theory, is to estimate controllability strategies, to figure out where in the brain those control strategies exist, and then how those control strategies may be explanatory of behavior or of symptomatology. But the second thing we can do beyond estimating controllability is to estimate the energy required for a state transition. So this is the second thing that I would like to talk about in this middle section. So in particular, what I want to say is that we can estimate what's called a minimum control energy. And I want you to focus again on the picture first, and then I'll walk through the equation very briefly. So here, imagine that the brain is sitting at a state x naught, which here is activation of motor cortex. Now imagine that if nothing else happened outside of this brain, it would naturally flow along this gray line to some final state x capital T. But what we want to do is actually understand how the brain can be pushed into a different target state, which isn't where it would naturally flow to, but where we would want it to go. And this is, for example, activation of visual cortex. Now, if we want to understand how this trajectory might occur and what kind of control inputs are necessary, we can write down an optimization, and in particular, I'm going to write down what an optimal trajectory would be between this initial state and this final state. And that optimal trajectory is one that's going to minimize two features. It's going to minimize how much input is injected into the system, and that is to align with biological constraints on energy consumption. And it's also going to minimize the distance of the current state x from the target state. And that is to ensure that this trajectory is as straight or linear or simple as possible, so that the brain doesn't move to some random spot in the state space when it shouldn't. So by minimizing these two features, we can estimate what inputs need to be injected into the system and when to enable this trajectory to occur, rather than this gray one, which would be the default. Now, that provides us with an estimate of not just the input into the system, but also the energy required to inject that input. And one of the questions that we had after we had developed this from a theoretical perspective is that energy that we are estimating related to biological energy, or is it just this mathematical quantity of energy that's interesting from a theoretical perspective? So is theory aligned with biology? And that is a question that Xiaosong He wanted to answer. He was a postdoc in my lab before he started his own lab in China. This is a paper that came out in 2022 in Science Advances. And what he did is that he asked whether the control energy that we can estimate using this mathematical approach, and this theoretically grounded mathematical approach, is related to glucose metabolism. So a biological notion of energy consumption. To address this question, he used FDG PET imaging to mark the glucose metabolism, and then he used individual specific diffusion imaging to estimate the regional control energy. What he found, I'm not going to walk through the data, but what he found is that there's a really beautiful, strong correlation between these two metrics, indicating that control energy, a mathematical quantity, is related to biological energy, particularly in the form of glucose metabolism. So there's a biophysical grounding for the theory that we are proposing. So with these tools in hand, we can ask many questions. We can ask questions like, which brain states require least energy to go towards? Or how much energy does it take to activate this cognitive circuit versus that cognitive circuit? Or are the brain networks in some species easier to control than others? Or could individual energetics be used to personalize brain stimulation? Or do these energetics explain individual differences in how we can learn brain-computer interfaces? Or how do I use these methods on my own data? So here are some papers that we've written over the last couple of years to answer some of those basic questions. But with that tool and formalization in mind, I want to move to the very last section, which is how can we use this notion of a network economy that has particular costs to brain state transitions to understand something important like mental effort. And here I'll again subdivide my comments into addressing 3 points. First, I want to discuss how higher cognitive load states, so those that require greater mental effort, are also ones that require greater control energy. So that's aligning our estimates of control energy with a sort of notion of cognitive or mental energy. In the second part, I'm going to state that development alters these energy requirements for executive functions generally, but which would include moving to higher cognitive load states. And then in the last part, I want to just mention that we can have pharmacological interventions that modulate energetic requirements in this network economy. So before I get into some of the data, I want to clarify terms. How am I going to use the phrase mental effort? I'll be using that phrase to indicate the engaged proportion of limited capacity central processing. So here what I'm showing is that we have this notion of mental effort that is correlated with a range of different variables. Most commonly, it's thought of as being correlated with task difficulty. If the task is more difficult, it will require more mental effort to perform and to engage in. It's also been shown to relate to task novelty. The more novel the task, the more mental effort we may need to engage. And third, it's related to a variety of different individual variables like age, neurotypicality or neuroatypicality, other dimensions of difference, etc. But importantly, there hasn't been a very clear notion of a identical relationship between mental effort and any of these functions. So mental effort is not identical with how difficult the task is, or how much motivation the person had, or how much attention they devoted, or how much control was engaged. It's related to each of these factors, but not identically. So how do scholars measure mental effort? They do so in a range of different ways. The first and simplest, perhaps, is by self-report. So you can simply ask the participant, how effortful did that feel? And they can mark, did it feel very effortful? Did it feel less effortful, etc. But along with that quantification comes all of the limitations of self-reported measures. A second way that we can measure mental effort is using autonomic response. So measures of physiological markers of autonomic arousal. So the more autonomic arousal, the more effortful that task may have been. There are also measures of neural activation that have been posited. So when two people perform the task in an identical way in terms of behavior with identical accuracy, reaction time, etc., but one uses greater activation than the other, then you can say that this person used more mental effort than this person because they reached the same behavioral levels, but this one activated more. A fourth way that has been proposed is to quantify the dynamical complexity of the neural time series. So when a time series has more dynamical complexity, it's proposed that the person has engaged more mental effort. So these are four sort of different ways of thinking about what mental effort might be and how it might differ across individuals. What I would like to propose is an alternative that can complement those previous ones, not to supplant them, but to complement them, in specifically thinking about what is the cost of orchestrating complex activity dynamics on this network. And I operationalize that in the context of network control theory. So specifically, I would like to understand what is the cost in terms of control energy for pushing the brain from where it is to a state that requires more mental effort. One of the places that we've engaged this kind of approach is in studying working memory. And this is work that was led by Urs Braun when he was a visiting postdoctoral fellow in the lab. And before he went back to the Central Institute of Mental Health in Mannheim, Germany to start his own lab. And what he did is that he focused on individuals who were performing a zero-back working memory task. So that just means they needed to report the stimulus that was currently on the screen. So that just takes focused attention. It doesn't actually take memory. And then the participants would move over into what's called a two-back working memory task, where they had to report the stimulus that occurred 2 moments ago. So they spent some amount of time in the zero-back condition, then in the two-back, then in the zero-back, then in the two-back, and they would move back and forth between these two sort of states. What ERS wanted to understand was how much energy did it take to transition from the 0-back state of activation to the 2-back state of activation? How hard was that? How much energy is required for that state transition, and vice versa? He was also interested in asking how much control energy is required to persist in either of these states. So to persist in the 0-back state, this attention demanding state, or to persist in the 2-back state, this working memory state. To quantify this transition energy and this persistence energy, he used network control theory. Here's the first piece of data from this study that is relevant for the current focus on mental effort. So this is focusing on transition energy. How much control energy is required to transition from the 0-back condition to the 2-back condition. You can see that here, with each data point indicating a different participant in the study. And then here's the control energy required to move from the 2-back to the 0-back. Importantly, what I want you to notice is that more control energy is required to move into the more cognitively demanding state, and less control energy is required to move into the less cognitively demanding state. What that indicates is that control energy is tracking with the mental effort of that transition. And in particular, that transitioning into a more cognitively demanding state will require more control energy. So that's about transitions. What about persisting in either of these states? Well, here's the persistence energy along the y-axis, and then here are the two conditions. So this measure, I'm calling this stability. That's the inverse, so 1 divided by the control energy. So when control energy is large, the stability is low, and when control energy is small, the stability is high. So here you can see that the 0-back stability is much greater than the 2-back stability. What does that mean? That means it requires less control energy to persist in the 0-back condition, the simpler task. And it requires more control energy to persist in the 2-back condition. That's the more cognitively demanding task. So again, aligning with our perceptions of the cognitive effort required for these two different states. Now, I also want to point out that there's significant individual variation here, and we did study that individual variation further and found that greater stability in this 2-back state was associated with higher task accuracy. So for those individuals that required less control energy to persist in the 2-back state, they performed better. And for those individuals that required more energy to persist in the 2-back state, they performed worse. So that's again consistent with the original sort of framing, but also suggests to us that individual differences in performance can also be explained by these control energetics. Now, in a final piece of data from this study, ERS wanted to examine the role of dopaminergic signaling in potentially altering the energy landscape on which the brain is moving, and therefore the energetic requirements for state transitions. He did that in two ways. First, he used a randomized, placebo-controlled, double-blind pharma fMRI study, where he either provided amisulpride, which is a selective D2 antagonist, or a placebo. And then he assessed the control energy again for these two state transitions. So here you can see the placebo in dark blue, and then individuals who received the amisulpride in light blue. And what we observed is that the drug increases control energy for both transitions versus placebo. So it's making both transitions more energetically demanding. Now, in addition to, and that is data in support of the role of dopamine in modulating or determining the energy landscape on which the brain is moving, and therefore the cost of brain state transitions on that landscape. The second way that he went about addressing this potential role of dopaminergic signaling was to use the same study design, but in patients with schizophrenia, who we know have marked dopaminergic abnormalities. They also have deficits in executive functions, including working memory, and they have alterations in structural connectivity, which are a key predictor of this control energy measurement. What you can see here is the data on the right-hand side, with the lighter color being healthy controls and the darker color being people with schizophrenia. What he observed is that the patients with schizophrenia require more control energy when transitioning into the more cognitively demanding state, and no effect when they come back down to 0-back. So there's a specific alteration in the control energy required to move into that cognitively more demanding state, again supporting the potential role for dopaminergic signaling in energetic requirements. Now, in the second part of this development of why the network economy is relevant for thinking about mental effort, I wanted to note that the network economy is changing as kids are developing, and it's changing at the same time that the mental effort engaged can be changing as well. So this is work that was led by Zhaizhu Qiu, who was a postdoc in the lab of Ted Satterthwaite. And what he did is that he focused on how much energy is required to activate the frontal parietal system of the human, which supports executive functions like working memory, but also complex decision-making, cognitive switching, task switching, cognitive flexibility, and inhibiting inappropriate behavior, for example. So in 946 youth between the ages of 8 years and 22 years of age, he asked how much energy is required to get the brain from a baseline of no activation to activation of this frontal parietal circuitry. So that's the state transition that we're studying. And each of these individuals has their own pattern of structural connectivity. So each of them has a different control energy required for this state transition. Here on the left-hand side is the first piece of data from this study. Along the y-axis is the control energy for that state transition. And then along the x-axis is age in years. And you can see a decrease or a negative relationship between these two variables, such that the younger kids require greater control energy to activate frontal parietal cortex, and the older youth require less control energy to activate the frontal parietal cortex. So that's interesting. It suggests that as the white matter networks are developing over this age range or in this developmental period, they are changing in a way that supports activation of frontal parietal circuitry with less energy. But we also wanted to know if that's simply a developmental effect or if it is more particularly related to individual differences in executive performance. And so that's what I'm showing you here on the right-hand side. Along the y-axis is executive performance as measured on a 2-hour cognitive behavioral battery. And then along the y-axis is the control energy. Again, you can see it after we've regressed out the effects of age. Again, you see a negative relationship here, such that individuals with greater executive performance are also those who require less control energy to activate the frontal parietal system. By contrast, those with less executive performance are those that require more energy to activate this system. So these two pieces of data provide evidence that supports the notion that the white matter network is developing to ease the transition to executive function, and that facilitates the frequent use of that cognitive process in older youth. Now, the last piece of data that I wanted to provide in this final section is demonstrating not just that development can be a force that changes the network economy, but that also pharmacological interventions are forces that can change the network economy. I already provided you with an initial piece of data in support of this in the study that used amisulpride that Urs Braun led. But I also wanted to provide this final or second piece of data that was led by Arun Mahadevan, who was a postdoc in my laboratory before moving on back to California. And what he did is that he focused again on schizophrenia, but in this case, he was particularly interested in relatives of people with schizophrenia. He was, was building on the sort of widespread observation that people with schizophrenia are marked by deficits in facial affect processing, and that's associated with abnormalities in GABAergic circuitry. This kind of deficit is also found in first-degree relatives of people with schizophrenia. So it's a really nice place to be looking for intermediate phenotypes. And his question was, could these deficits be explained energetically in a network economy? So he took data from as participants were performing a facial affect processing task, so identifying emotions in faces. And he also took individual differences in their white matter tractography. From both of those pieces of data, he could estimate the amount of energy that was required to persist in emotion identification versus not persist in emotion identification. And what he found is that alprazolam, which is a GABAergic agent, increased persistence energy in relatives of people with schizophrenia, but not in the controls. And that was particularly during the processing of threatening faces or identifying emotions in the threatening faces. So again, that suggests suggests that there can be pharmacological interventions that alter the energy landscape on which the brain is moving, and thereby the cost of state transitions in the brain that are supportive of cognitive functions. So with that, I would like to summarize what we've done over the last couple minutes and just give one quick note on next steps, where we're going from here. So first, to summarize, we started by mentioning that the brain's structural network constrains the flow of activity, thereby influencing the types of computations and functions that can be performed. Then I noted that network control theory models those constraints, those structural constraints, and provides an estimate of the energy required for brain state transitions. In the final section, I focused on cognitive effort as a place where we can start to study this network economy using notions of control energy. I noted that greater control energy is required for transitions into more cognitively demanding states, and that it can be modulated by pharmacological interventions. Why, and all of these pieces sort of come together to help us to quantify the economy that exists in the brain that can be impacting how the brain is functioning. Importantly, by estimating these transitions and persistence costs, we can assess which brains or brain regions are wired for more economical transitions and in what contexts. So where do we want to take this work from here? We want to take it in three directions. In the first, we want to be able to use this approach to identify deviations from a normative trajectory of network economy. And this is work that's based on ideas that Lyndon Parks published in a recent translational psychiatry paper, again, before he moved on to building his own lab at Rutgers. And he focused on studying the normative trajectory of brain development, and then also by indicating when those trajectories are diverging from the normative trajectory. What I would like to do is to understand how the brain diverges from a normative trajectory of network economy and how that may impact cognitive processes. The second way that we want to expand the work that I've mentioned today is by understanding how those deviations from a network economy impact the cognitive systems that are symptoms that are observed transdiagnostically across a range of different conditions. And this is based on ideas in a recent review article that I co-authored with Duncan Astle. It just came out, I think, like two months ago, maybe, in Neuroscience and Biobehavioral Reviews. And the third and final way that we want to expand this work is by not only studying normative network economies or non-normative network economies, but also by understanding how the network economy inside the brain may relate to economies in cognition, in behavior, and in environment. So I want to acknowledge that the brain doesn't exist in some isolated dish. It exists in connection with a mind, with a body, with behaviors, in an environment that has social influences and constraints of its own. And that in order to understand the economy in which we work, we have to be understanding multiple systems that are interconnected with one another. So with that, I would just like to thank you all for listening. I would love to take any questions that you might have. Yeah, okay, so the question is that the control input is often framed in the context of an exogenous signal, but what we're doing is that we're applying it to try to understand intrinsic signals. And so that there's a disconnect there conceptually, I think is what you're pointing out. So, and can I talk about it? Is that just kind of, yes. Okay, great. So yes, I think about this approach as a way of modeling both extrinsic or exogenous input and endogenous input. Probably the easier place to start is exogenous input. So in a bunch of work that I didn't talk about today, we do study the impact of stimulation on neural dynamics, and we use the control models to model the stimulation to a particular area and how that impacts the activation in that area and other areas. So that is a line of work that exists in my lab, but that I didn't talk about today. What I did talk about is when we're trying to use this same approach to model endogenous dynamics and endogenous control properties. So the way that I think about it is that the brain may be existing in a particular dynamical state, and then there are inputs that are coming in mostly from tasks. So inputs into the visual system, inputs into the auditory system, inputs into our sort of whole body physiology that then come into the brain. So there are multiple inputs that are coming into the brain. And then those inputs are being transduced or transformed into control signals that enact particular cognitive processes. And that's the control signal that I'm really interested in understanding. And that's the one that would support things like executive function. Yeah, I think that, so what we can do is that we can take individual data before the frontal parietal system is activated. We can say that's the first state. And then we can take this state after the control system has been activated. We take that as a second state. And then we say, what is the control input that we would model, like in simulation, that would be required to enact that state transition? And then we can say that estimated input is what we think the frontal parietal system is doing. So it's a way of modeling what intrinsic control inputs are there, but that may be occurring at too short of a time scale for us to estimate with fMRI. But maybe another piece of data I want to add here is work that was led by Eli Kornblath in my lab. He was an MD PhD student. He's now a resident. What he did was, when we're trying to model state transitions that we observe during the resting state, we can model them best when we allow inputs into all regions of the brain, because all regions are kind of being activated and all involved in the movement of the brain across states during the resting state. But when we are focused on a task that provides significant visual input and where responses are dependent on the visual input, the only way that we can model those accurately is by only allowing control signals into the visual system. So this provides a different way for us to think about understanding task demands, because we can say, given what we observe in the dynamics, we can estimate where would we need to be putting input for this to happen. And that allows us to be more concrete in modeling the sorts of input that the brain is receiving to allow that cognitive task to be performed. So the question is, can we think about economy in our social economy, so that where we exist in the context of social inequalities as an input?
No, I think in the same way in the brain. So rather than just thinking of economy as cost as one factor that affects the transitions, could there be differences in the states of the economy, which affects the mental health more?
Yeah. Okay. So is it possible that the system can exist in different states of economy, and that those may be impacted by socioeconomic status or social inequality or other things, and then those may be explanatory of differences in brain function, specifically in the context of mental health? Yes, I think that the economy that I am describing is one that's really focused here on kind of thinking about what economy looks like for a complex network inside of a neural system. But I do think that there are economies that exist in the environment. We know that there are financial economies and social economies and value all sorts of things that then may themselves be impacting how the brain is developing or how the brain is functioning or what states the brain can reach. So yes, I think that what I'm describing is one slice of this multi-scale system, but I do think that the approach is relevant across scales. And maybe just to clarify what I mean by the approach is that I think that each of these scales has network architectures that constrain how the system can move, and that we need to take into account those network structures to understand energetically where we can push and how we can push. And so when we're thinking about interventions for mental health, I think those could occur in the brain, maybe if that's where there's most malleability, or they could occur in the environment if that's where there's most malleability. We don't really know in many cases. But that this approach of examining the network architecture as a formal constraint, I think is one that's important and relevant across the scales. So just to repeat the question, you're mentioning that in some mental health conditions, they may be characterized not necessarily by differences in the state transitions, but by differences in the overall level of activity. So sometimes just overall less activity, or sometimes overall more activity. And the question is, how might you examine the network economy in those individuals where the state transitions are not necessarily altered, but the kind of the baseline is different? So where the effort has surpassed some sort of threshold such that they can't or they don't activate to a particular degree that would be necessary to perform the task in a particular way. Yeah, I think that's really interesting. I think that I wonder if there's a multiplicative factor that we could add, where it's not just about how much energy we would predict for that person based on their structural connectivity, but also about how much energy it practically feels like for them, based on additional factors that may be related to the dynamics of individual regions. This question, so I don't have an exact answer to your question because we haven't examined that. But I did want to mention one caveat to some of the work that I've shown and a way that the field is moving to address the caveat. And I think it's relevant for the issue that you're raising. So in particular, all of the regions in the network model are treated as the same. So they are all units that have some activity, and the activity can flow to other units, but there's no distinction in how excitable each region is or how easy it is to inhibit a given region. There's nothing special about all of the regions are treated equivalently. But that's not how the brain works. We know that there are some regions that are more excitable and others that aren't. And that would have a big impact on how they can push activity through the rest of the system. So to address that limitation, there has been work by other colleagues in the field that have used estimates of neurotransmitter levels across different regions of the brain as an important marker that determines how responsive each region is to exogenous input or endogenous input, how excitable those regions are, and then how easy it would be for them to push activity out into other regions through existing tracks. And the way that that is done is by taking the B matrix that I mentioned when I showed you the original equation for the model of dynamics, and along the diagonal, instead of putting ones where each region exists, you put levels of neurotransmitters where each region exists and that you think are important neurotransmitter levels that may alter the excitability of the area or its receptiveness to input. So I think that that's one important way of considering additional biophysical constraints on the dynamics that may be impacted in many mental health conditions, and that may be explanatory of the distinctions in different baselines, the height of activation differences, which states can be reached and which can't be. So that's one way of adding more biophysical realism to the model that I'm really excited about. No, I haven't. But so the question is, I gave some examples in development, but could there be effects in aging as well, where controllability may be altered or the control energy required for certain state transitions might be altered? I think that is definitely something that I think is happening, but is not a place that we have done any studies. So I'd be really excited for anybody who wanted to do that study. I'd also be able to help, willing to help in any way. I want to mention, actually, this is a good time to mention that Lyndon Parks wrote a protocols paper, so was just accepted in Principle to Nature Protocols, where we explain how to use the tools. We have, optimized the code so it's really flexible and easy for others to use, and we walk people through examples really carefully. So that is an effort to make these approaches really readily usable for anybody. And yeah, so I would love to study this in aging or have somebody else study it in aging. I think that would be great.
So thanks so much for visiting us and for such a wonderful talk.
Thank you for having me.